So here we have a ribosome. It's a two-part organelle, and the two parts, hold on, you want to take notes on this. The two parts are the large subunit and the small subunit. Someone got paid big bucks to name those. But the large and small subunits, what they do is they float around in the cytoplasm unconnected. They only come together as an assembled ribosome when they're ready to sandwich some mRNA between them and begin that process of translating a protein. So let's look at the structure of ribosomes, which contain our RNA. The small ribosomal subunit has what are called mRNA binding sites. These are places where the small subunit will clamp down upon the mRNA and secure it to itself. The large ribosomal subunit has what are called tRNA docking sites. I think of these docking sites kind of like stages on an assembly line. And there are 3 stages on this assembly line, or docking sites. Docking site A is where tRNA molecules first kind of find their way to the large subunit and enter it. They are then moved over to docking site P, which is where they actually complete a peptide covalent bond between amino acids. And then once they're done with making that bond, they move into docking site E, where they prepare to exit the large ribosomal subunit. Of course, as one tRNA is moving through these 3 docking sites, there are other tRNAs moving through them also in order. So let's see what a tRNA is. Before we get any further into the machinery like ribosomes and polar covalent bonds, let's meet the translator. tRNA is a molecule that can both interact or bond with a nucleic acid and an amino acid. And this picture here on the left is a pretty realistic picture of tRNA. It's just a strand, right? tRNA is just a strand of RNA, or it's just a sequence of nucleotides, and it seems to have a messy shape, but it has a couple places on it that are very important. On tRNA, there is a site that is called the anticodon. And here we go right here. The anticodon has a sequence on it. And let's just say, just for argument purpose, let's just say that the sequence of nucleotides on the anticodon is UAC. And that matches up with a corresponding codon on mRNA. Codons are on mRNA, anticodons are on tRNA. Also, the tRNA has a site on it that will match with an amino acid. And you're going to say, Amy, which one of the 20 amino acids will it match with? Well, that's predetermined. But each tRNA has a specific amino acid that they only connect to, just one. For example, a tRNA that has the anticodon that's UAC will always carry an amino acid called methionine that we abbreviate as MET. Always UAC. If you find a tRNA that has UAC on the anticodon, methionine is the amino acid that is attached to it. I was just about to say that this picture on the right is actually a more realistic picture of tRNA, kind of showing its bulk. I don't know, though, that I'm looking, I feel like the picture on the left, I'm more able to understand it. The picture on the right is just a big globular protein. All right, so this is tRNA, and it is like the decoder ring, like a translator, that something knows what is called the genetic code. This is the genetic code. This is a genetic code represented as a dial or a circular concept map. There are also genetic code maps that are rectangular. I encourage you to take a look at some of those. In looking at the process of translation, though, it occurs to students that there seems to be a very specific match from a codon on tRNA to an anticodon on, a codon on mRNA and an anticodon on tRNA, and then to the amino acid in the protein. Yes. In fact, this relationship is set in stone, and it's reflected in this very strange picture here. There are, like I said, some tabled or grid representations of this, the genetic code. You might notice here that A, G, and C are present, but thymine is not present. This picture is restricted to representing only nucleotides that are present in RNA, and that's A, G, C, and U. Actually, these are going to represent nucleotides in our mRNA that were made in transcription. What this picture does is connect each codon, or a 3-nucleotide sequence of mRNA, with a specific amino acid. Let's do an example. Let's say that on the mRNA that you have a sequence of amino acids of A, U, and G, let's say, and that would be on the mRNA, and this would be a codon. So let's start at the middle of this ring of the diagram, and let's choose A. And that's in the red on the bottom left of the inner circle. Okay, let's move outward one circle, and let's choose U, which is in green, almost at the bottom of our circle. Okay, let's move outward one more ring, and let's choose the G that's in blue. And the amino acid that corresponds to the AUG codon is methionine. And some of you might be thinking, but Amy, that matched to that other codon that we looked at on tRNA. Oh, it did not. Our tRNA that matched with this codon was UAC, which strangely uses the base pairing mechanism to match with our AUG codon. Let's do another one. Let's do GGG. And pause this video and do that right now. Did you get SER? Did you get SER? No? Good, because that's not it. I just didn't trust you to pause the video quick enough. So the GGG codon on mRNA would match to this amino acid up here called gly or glycine. There's also an amino acid that matches with UGG, and that would be tryptophan, which is the sleepy stuff in turkey. One more. Try another one for yourself. UAA. And UAA along with UAG, both of these are called stop codons. They don't correspond to an amino acid. What they do is they make the ribosome spit out the mRNA and stop translation. So this decoder ring here, it's the English to Swedish dictionary that I need to translate my Swedish cookbook. What's cool about this is that if you can see, even though one codon will have only one amino acid to match with it, one amino acid matches to a few different codons. And this allows flexibility to create an unlimited, as currently known, an unlimited number of proteins for life, even though we have only 5 different nucleotides and 20 different amino acids. Let's look at the process of translation. We can see here I've got the small and the large subunits of the ribosome coming together, and they're sandwiching that mRNA between them. And here is our mRNA. The small ribosomal subunit finds the codon AUG, and it makes sure that that codon is locked into, here's AUG, they make sure that that codon is locked into that middle bay or that middle unit of the large ribosomal subunit. The large ribosomal subunit also finds a tRNA that has methionine, MET, attached to it as an amino acid. This is how every single protein begins, because there is only one start codon, and it's AUG. This also means that methionine is the first amino acid on every single amino acid chain. However, during folding and splicing, methionine might be cleaved off of the final protein shape. But with the mRNA bound to the small ribosomal subunit and the tRNA bound to the large ribosomal subunit, we can now begin the process of elongation of translation. Isn't it when you first look at these pictures that you look at them and you're like, these are all the same picture? That's how I feel sometimes. In this first picture on the left, we can see another tRNA, the pink one, coming over to the entry site on the large ribosomal subunit. And this tRNA has a codon of GUC, which matches, this tRNA has an anticodon of CAG, which matches with the codon of GUC on the mRNA. Once docked into the A site, the amino acid from the P site is bonded to the amino acid over there using a polar covalent peptide bond. Take a really good close at this diagram and confirm this codon on the mRNA, confirm the anticodon on the tRNA, maybe go and refer to the genetic code and see what this specific amino acid should be in our example. The second picture here shows you the first and the second tRNAs kind of moving over one docking site. I feel like that's like rotating in volleyball, right? Rotate. So the tRNA that now doesn't have an amino acid, it moves over to the E docking site, getting ready to exit. And the tRNA that has the two amino acids attached to it, it now moves to the middle or the P docking site. This leaves the A docking site open and capable of attracting the next tRNA. The next tRNA has an anticodon of AUG on the tRNA and a codon of UAC on the mRNA. So these little tRNAs, they're actually floating around in your cells all over the place, and they've been produced for you by the breakdown of DNA from other organisms that you eat. These tRNAs all day long, all they do is go to your digestive system, pick up an amino acid, and then go to some cell and drop off the amino acid like they're doing here. Then they go back to the digestive system to pick up an incoming amino acid and go back to a larger ribosomal subunit and drop it off. So tRNAs, they're like taxis for amino acids. That's kind of what they do. The third picture here, it shows you our very first tRNA leaving the large ribosomal subunit without its amino acid, right? It's going to go back and pick up another one. And it leaves the other two tRNAs to now move over into the P docking site. And so we continue this process, matching tRNAs to the mRNA, forming peptide bonds to be the amino acids, and moving right to left, right to left, right to left in this picture. I know, bear with me, we're almost done. So the last thing that happens in translation, it's a process called termination. And this is when the text of one of those stop codons in the mRNA hits one of the binding sites. The stop codon will trigger the amino acid chain in the P docking site to be released from its tRNA. Here is a stop codon right here, UAG, and what it's doing is it's basically saying, no, there are no more tRNAs that will be coming over here. We are stopping this process of translation. And so now we have this amino acid chain, but in and of itself, that's not finally a functional protein, right? Remember, proteins have to have levels of structure that reflect the folding and the stabilization of those folds by hydrogen or intramolecular bonds. However, the folding of proteins is done by other proteins. And that's a process for another PowerPoint. But now, knowing that proteins are folded by other proteins, you should be asking what folded the first protein. Okay, I just want to address this last picture here, where you see everything breaking apart, right? The large and small ribosomal subunit get broken apart, and they go and find another mRNA. The mRNA here, it's a disposable copy. So it gets degraded by enzymes that are constantly in the cytoplasm. And then here we have our new amino acid sequence that will go on to be folded.